morning, everyone. Welcome to today's Catalyze seminar session, How to Source, Vet, and Oversee Medical Product Development Contractors. My name is Patrick Ahern with RTI, and I'll be helping out with the virtual room today. Please note when you log in, your microphone and web camera will be automatically muted. If you have any questions for our presenters today, please submit those into the Q&A window. And if you happen to run into any issues with the Zoom, you can reach out to me in the chat window. Towards the end of our session, we will have a brief evaluation poll, so please do take a moment to provide your feedback at that time. So Marissa, I'll go ahead and turn the meeting over to you. Thank you.
Great, thank you so much, Patrick, and thank you everyone for joining today. Welcome back to our Catalyze webinar series. As many of you know by now, my name is Marissa, and I work on the RTI side, and I'm so excited to introduce today's webinar, which will cover sourcing, vetting, and managing medical product development contractors. Our presenters today are two of our technical project leads from the Catalyze Coordinating Center, Willie Salminen and Greg Gatto. Willie's 20+ years of drug development experience includes regulatory, non-clinical, clinical, and CMC aspects of the development process. And Greg brings extensive experience in research pharmacology and directing scientific research programs from target validation to first-time-in-human studies. So with that, I hope you enjoy our webinar, and Willie, take it away.
All right, thank you, Marissa. Well, it's a pleasure to be able to present this information today, and I'm going to try to come at this from a more practical standpoint and hands-on, what you need to do to, find labs that can do the different type of medical product development work that you need. And basically, it's really ensuring this efficient, timely, and high quality studies and data for regulatory submissions is really the focus and goal of this short course. So you can go to the next slide. So some of the key goals here are demonstrate the importance of selecting what I call fit for purpose contractors. So you may have a contractor, you may be more price sensitive, you may be more quality sensitive, or maybe a hybrid of the two. So it's trying to figure out what CRO or CDMO fits what you need and what you're looking for. And in terms of definitions, CRO, I'll be using throughout the presentation, stands for Contract Research Organization. And a lot of times they're going to be ones that run your non-clinical animal studies or clinical trials, so your human studies. Contract Development and Manufacturing Organization, or the CDMO, and there's other acronyms that you'll hear too. They're the ones that are more making the drug for you or the medical product device. I'll delineate the critical role of being actively involved with your outsourced activities. I always say never just hand the keys over to a CRO or CDMO, and I'll explain why later. Because that's just a recipe for disaster. So you really want to be actively involved with any kind of activities that you outsource. And they'll provide an example of the steps in selecting and contracting outsource activities. It'll be focused more on non-clinical outsourcing, but the steps and sort of overall process will be applicable to any kind of interactions you have with a CRO or CDMO. Okay, you go to the next slide. And then you can go to the next one. So in terms of why it's important to be actively involved, I'll just give some, high bullets level things on the next slides here. So first of all, your study is very important to you. You have more personal investment in the success of the product than the CRO or CDMO does. So yeah, the CRO wants your product to be successful because they're going to get paid, they're going to, their reputation's on the line, but you are personally invested in the success of your company or your product, which can either feed your company or just, publications or whatever you're looking to do with it. Your reputation is linked to the success of the study. Not necessarily the results, but the quality of the study. So you can control the quality of the study by being involved, but you can't necessarily control the results because the results are, drug dependent, device dependent, but you do have a big role in playing in terms of the quality of what the study ends up being. You want the highest chance of regulatory acceptance. This can apply to good laboratory practices, which are GLP for your non-clinical animal work, good clinical practices, which are the GCPs for your clinical work, and then good manufacturing practices, which are the GMP for your device, for your drug development in that particular example. Then you have scientific quality, which can be separate from the GLP, GCP, and GMPs, and I'll explain that a little bit later. So the scientific quality would be the design, data integrity, and the results, and whether that meets the regulatory requirements that you're looking to meet. And then you, the granting organization, and the company, or any one of these mixes, are relying on a successful study. Again, you can't guarantee the results, but you can influence the quality. So a lot of times, using drugs as an example, I mean, if a drug's toxic, it's toxic. You can't make it safe, but you can make sure that the study is run well. Okay, so you can go to the next slide. You know the product best. So you're going to be working with contractors that don't know your product. You have a lot more background information on your product. In terms of CMC, which is your manufacturing, you know the synthesis process, even if it's just at a lab scale, you know potential hurdles, you know the potential stability and desired formulation. Or if you don't, at least some of the stuff can feed into, you know, the discussions you have. For non-clinical, you know most likely the type of study design you want, dose setting, ballparks, you know, that you're looking at based on probably preliminary data that you may have, any specific animal welfare issues or animal strain requirements you may have. For clinical, you know, you'll have hopefully some information on dose setting, the study population you're looking at, and potential safety issues that the CRO may not be aware of, and you'll have to feed this information into the CRO so they can provide, you know, interactive designs with you. You know the target product profile and your development plan, so you can determine which CRO or CDMO provides the development services that best align with your development plan. Again, just as a simple example, are you focused more on quality, or you're focused more on, you know, speed to market or low cost? And again, it's a balance of all these things that you got to look at. In terms of physical chemical properties, for a drug particularly, you will know those things. You can provide that to help with possible formulation development, analytical approaches, any kind of local irritation or worker safety issues that may be an issue with handling the raw drug itself. And then you can play a key role in helping the CRO or CDMO be efficient, cost-effective, and successful. So if you just tell them, hey, we want this study run, and you give them almost no background information or very limited and let the CRO run with it, they're going to be asking you tons of questions. They're going to have to be doing a lot of legwork themselves. And so the study costs could go way up on you. Whereas if you feed them a lot of information up front, you can make it not just, you know, more cost effective, but, you know, quicker and higher chance of success. And like I said, I'll outline later how many questions need to be answered both before, during, and after the work is done. Okay, you can go to the next slide. So this thing I really like to highlight, because if you never worked with a CRO or CDMO or ever worked in that environment, you may not be aware of this. CROs and CDMOs are typically very busy places. They're very exciting places to work at because there's a lot of stuff that's going on, a lot of cool things that they're working on. However, they are very busy. A typical study director at a CRO is going to have a whole list of studies that they're typically overseeing. And so I hate to say it, but the squeaky wheel usually gets the grease. I mean, you don't have to be mean about it, but if you can do it in a very nice way. But if you keep on, you know, just making sure the study director understands, look, I'm going to be on top of this study. I want updates. I want a good communication. I'm not just going to let you run the study, you know, without keeping me informed. That's really, in my opinion, the best way to interact with a CRO or CDMO, so they understand that you will be keeping an eye on things. Again, you don't have to do it in a mean way. You can be very positive about it, and just so they understand that you will be a client that's involved versus a client that's going to be very hands-off. They're obviously going to pay more attention to you. There's a typically pretty high employee turnover at CROs and CDMOs. So you may be interacting with people that are just getting up to speed and may not have a lot of experience, especially people that are maybe doing the actual work. That's typically where a lot of the turnover is. So you just want to make sure that, you know, you're keeping an eye on things to make sure that people, even if they're inexperienced, you can help guide and just make sure things are being done right. One thing that people get confused on is in terms of GMP, GLP, and GCP compliance, is that these are not scientific integrity. It's scientific integrity, or it makes a sound study, but it doesn't guarantee a high quality study per se in terms of regulatory acceptance or meeting scientific requirements. What these things do is help ensure data integrity. They help ensure that FDA, if they come in and review your work, that they can reconstruct what was done. So it's really good at reconstructing what was done, but doesn't necessarily make a good study. And I'll provide some examples later about that. And then for protocol amendments, you'll be making changes most likely. It's rarely if a study goes as planned according to the protocol. You're usually making little tweaks along the way, whether it's an amendment or even a deviation that leads to an amendment. But you can play an active role to make sure that everyone is aware of the changes, because a lot of times the protocol be amended, you get a new tech crew come in through the weekend, and they're not aware of the amendment possibly, and they're doing procedures that are against the protocol, and you get a deviation, which doesn't look good. Okay, so you can go to the next slide. And then you, the granting organization, company management, or mix of these will likely want timely and reliable updates as the study's ongoing. So pivotal studies can make or break a drug development program or company. I mean, I worked with companies that they're a single product company. They don't have anything they have investment funds coming in. That is their life. If they don't have that study successful or the drug successful, the company is going to go under. And so that when you have pivotal studies where it's an animal study, clinical trial, you'll likely need to provide continuous updates, for studies like that. You'll want updates from the CRO or CDMO. You'll want to do some remote monitoring of activities, studies, or data. And the CROs make this easier and easier nowadays. You can get access to their online database, real-time data collection, especially if they're using electronic data capture systems. And then, of course, going on site to monitor activities, the studies and data is actually always very helpful. COVID threw a wrench in the works there, but things are opening up more and more where you can go back on site and, look at the SOPs, go look at animal dosing in the case of a non-clinical study, go review any kind of paper data that they may be using. Because even if they're doing electronic capture, there's always paper data that you're going to be wanting to look at too, that they'll be capturing. Okay, you can go to the next slide. Okay, I thought this would be a good section to highlight where issues myself and others have come into where we've run into problems, and sort of highlight why actively being involved with your study is a good thing. Okay, so you can go to the next slide. So in terms of, and I'll break this up to non-clinical, clinical, and manufacturing. So in terms of the non-clinical, which is your animal work, I had one study, which it was a GLP rat oral gavage carc study. So you're looking at cancer development over, a couple year, 2-year period in rats. There was a very senior study director, and they were selected on purpose because they had a really good reputation in the field. So it was sort of like, okay, pick this guy because he knows what he's doing. However, so he was so senior that he really relied on other people to do the actual work, even stuff that he should have been doing as the official study director under the GLPs. So he literally did not go back to the study room or review raw data for 6 months. Again, he was a really high level person, which I guess theoretically I can understand that, but as a study director under GLPs, he needs to be doing specific activities for regulatory requirements, and he did not fulfill them. So that was a major issue on that study. There was another study in the middle here, a GLP segment 2 rabbit IV teratogenicity study. So this is the type of study. A seg 2 is where you're looking at birth defects such as like cleft palate and things like that. Rabbit study. There were a lot of animal deaths and injection site related reactions that the study director never mentioned and were not identified until an on-site monitoring visit. So that's the last thing you want, because if you are notified of these deaths and you're notified of injection site reactions, you can take measures to reduce the dose, figure out alternative methods of administrating the drug. Once you go do an on-site monitoring visit and say it's weeks down the road, it's too late, and then you may have a failed study, whereas you could have actually had a successful study if you got it early on or caught it early on. This last one was super interesting because it was drug exposure in control animals. So usually in control animals, you don't want to see drug exposure. That's going to be almost a failed study, depending on the exposure levels. And this one was, I say it was very well designed. It was a complex study because the way the animals, they had to be randomized. Usually you'll have one rack of animals in a room that are a control group, a separate rack's your low dose and separate rack, mid dose and high. The way the regulatory requirements for this specific study were is that they had to be randomized through racks. So you had a mix of a control, low dose, high, mid dose, and high dose on the same rack. And the techs did not know what the treatments were. And we had protections in place where the techs, between touching animals, they changed gloves. And just because they didn't know if they're touching one animal that was a control group and then going to a high dose or vice versa. And so that was theoretically preventing cross-contamination because it was a topical product. You could get it on your gloves and transfer it to the control animal. What we didn't think about was the animal techs that were actually changing the cages and cleaning the cages at night. They weren't changing gloves because they weren't aware of it, and we didn't even think about it. So we got control exposure in the control animals because they would touch a high dose animal, go change the cage in the low dose and a control animal. As they're pulling the animal out, that drug transferred over. So again, you run into things like this. In hindsight, we should have figured it out up front, but you run into these things and you try to figure out it the best as you can sometimes as it happens. Okay, next slide. There was another one where a study director was changed during the middle of a 6-month repeat dose rodent tox study, and technician on top of it was very high at the CRO. So there was a ton of dosing errors. Some key study functions were skipped, and particularly after protocol amendments and on weekends, because like you said, the techs on the weekends are sometimes different than techs during the week. And they weren't, since there was a new study director, they weren't keeping updated to the technicians saying, hey, you need to pay attention to this new study amendment that we made. So there's just a lot of errors on this study. And that's where your active involvement is really key to make sure that this stuff doesn't happen or is caught early. And this last one was really interesting. This is actually a study being run overseas. It is really trying to understand who is conducting study functions and really delving into it. So if it's a study, if it's CRO you're used to working with, usually you'll know who's doing what and who's qualified. This was a new CRO, it was overseas, and it was lucky that we dug into who was actually conducting what study functions, because during a teleconference, it was determined that on weekends, security, so this is site security people that were patrolling the facility, they weren't trained scientists, they weren't trained technicians, they weren't trained under the GLPs. They were going to go and do clinical observations on the animals on the weekends. And we're like, that can't be. They have to be trained under GLPs. They have to be documented. So it was almost a little funny, but luckily we caught it before the study even started. So, okay, you go to the next slide. In terms of CMC, some issues that we've run into in terms of synthesis, I was trying to get some prodrugs made, and theoretically it was an ester formation, so it was theoretically a very easy synthesis route. However, there were multiple published routes for the synthesis process, and the CDMO that we contracted with used the wrong publication to develop the method, and that was our bad because we weren't keeping on top of what they actually were going to use, and the communication process was very poor. Also, the CDMO used our 100-gram allotment of the starting material in the first attempt with the failed synthesis, whereas they really should have started off at 1 gram. But again, that was our fault for not being more actively involved and telling them, hey, start off 1 gram and making sure you use this process. And so we wasted 100 grams, a lot of money, and had to start back over. In terms of stability, you can have we've run into issues where stability studies on toxicology batch drug substance did not cover the duration of the tox study. So you sort of got to scramble and reanalysis the tox study batch after the study's done and keep your fingers crossed it is stable and versus stability up front. It can also apply to GMP batches for clinical work. You want to make sure you have adequate stability coverage typically. And then this last one was interesting where we had a study in dogs that we were doing it was an oral mucosal spray, and there was some GMP stability work that indicated potential precipitation issues during storage, but was for various reasons we decided to proceed with the dog study. Midway through the 6-month dog study, actually it was like a 9-month dog study, The drug product, even under controlled storage conditions, started precipitating out. And of course, precipitation in oral mucosal spray was clogging the pumps. And it was basically ended up being a failed study. And we had to start over and we had to redo all the, stability and GMP manufacturing there. Okay, you can go to the next slide. Okay, I'm going to skip the first one and just go to the next one in the interest of time. And so, oh, I'm sorry. No, go back to the previous slide. I meant the bullet. So, okay, thanks. In terms of component selection for this example, there was an IV tubing set, and there was a glue that needed to glue the needle to, or the plastic hub for the needle onto the tubing set. And that glue that was selected ended up being cytotoxic. So that was an issue in terms of trying to get that. We had to scramble and try to find a new glue that was not cytotoxic. So because it was a combination product where the drug was being delivered through a tubing set, and they were selling the drug plus the tubing set, which is a combination product, device plus drug. And so the cytotoxicity was an issue for getting the product registered. Okay, next slide. In terms of clinical design, we had one issue where the inclusion-exclusion criteria were very restrictive for this clinical trial. And we had to go back to FDA to discuss with them about broadening the inclusion and exclusion criteria so we could recruit, more reasonably. Instead of being several years, we could redo it over 1 year. It was that bad, the restrictions. But those restrictions FDA initially required, but we're able to convince them to change those. Sample collection and storage can be an issue with clinical trials because they're not equipped like a lot of non-clinical CROs to handle some unique samples, because a lot of times it's out in doctors' offices and stuff like that. And so we had one study where it required special processing for the PK pharmacokinetic samples. And it was really difficult for a lot of the sites. And so we had a lot of errors with sample collection. The last one was actually really bad, where we got the PK profiles for drug exposure on a clinical trial. It was being conducted overseas. We did not do any monitoring visits. And that was a huge mistake because when we got the PK data back, supposedly it was different patients, but their PK profiles lined up perfectly, which there's no way that can happen. So basically it was data that was being made up, and we had to really revisit, the quality of that study and data integrity. Okay, so the next slide. So in this section, I'm gonna focus more on outsourcing a non-clinical animal study versus the manufacturing, the GMPs, or the clinical, the GCPs. So this will focus more on the GLPs and non-clinical studies as an example of the overall process that is, seems to work well for keeping involved in the study. Okay, next slide. So first is selecting a lab to run a non-clinical study. You can go to the next slide. So the location of the CRO is an important consideration. In the US, most CROs are going to be very familiar with FDA GLPs and in the inspection process. One thing that's interesting that's a lot different than some countries outside the US is that in the US, labs are not pre-certified as GLP. It's up to the lab to do GLP work, and then FDA will inspect to make sure the work was done under the GLP. So there is no pre-certification of a lab doing GLP work. So any lab can say, I'm doing GLP work. Once that study is done and FDA inspects them, then it'll be determined whether that study was done under GLPs. Whereas in overseas, in quite a few countries, the lab has to be pre-certified GLP compliant, and then they do get inspected after the fact. But in my opinion, the inspection after the study is done is actually lighter because it's they go in with the assumption that if the lab's certified GLP, the work will be done under GLP, so they don't have to do as hard of an inspection. I personally prefer the US process. And then also you just need to be aware that if you are experiencing an FDA audit, they are going to follow the US process and do a really hard audit on your study once they go to the CRO to look at that study, if it's being done under GLPs. Of course, it's easier to, if you're located in the US, it's easier to monitor the studies in the US, similar time zones. The main downside is the cost of the study can be higher than outside the US. Okay, next slide. So outside the US, they may or may not, the CRO may or may not be familiar with FDA GLPs and inspections, depending on how much experience they have. And like I said, I mentioned the GLP certification that some lab countries do. More difficult to monitor, widely varying time zones, but the pricing, the pro is the pricing can be very good relative to the US in terms of that. Okay, so next slide. Then you got different types of laboratories that you can possibly work with. The first one is academic, and I'm going to talk about purely academic. So it's a university that doesn't do GLP work, because there are some academic facilities and universities that have GLP labs associated with them. So that would be a little bit different. So that would be more aligned with, I'll be showing on the next slide, but I'm talking pure academic where they don't do any GLP work and no experience doing that. So typically non-GLP, it can be very difficult contract negotiations just because for a variety of reasons, publication rights, things like that. It can, in the IACUC, your Institutional Animal Care and Use Committee, which is has to authorize each study that is conducted in the US. They can be difficult to work with in the academic setting just because animal welfare issues are a different level than with private typically. Then you got government labs in this. So I used to work at FDA, and we had a big research facility I was working at. So we did a lot of GLP work and non-GLP, but we couldn't really do commercial work with commercial clients. Typically, the work was conducted for consortia or internal work or independent research agreements. So it was more internal to FDA, internal government work, or consortium where you're trying to get something for the public good out there. So really not in it for a profit type work. So the government facilities can be off limits on a lot of stuff that you may want to do there. Okay, so next slide. So the ones I'll focus on is the ones that you'll typically be interacting with, and these are the CROs that I mentioned before that run GLP studies. They can run both GLP and non-GLP. A facility that runs GLP easily does both. And I got that question the other day, can a GLP facility run non-GLP and vice versa? So a GLP can run non-GLP studies, no problem. They just don't do the GLP part of things. The non-GLP lab may or may not be able to run GLP. I mean, they can say they run GLP, but you need a lot of infrastructure in place, a quality assurance unit, appropriate, training, a lot of infrastructure is involved to get up to speed to run GLP studies. There can be much easier contract negotiations and IACUC approvals. They're typically very responsive, and then they have established GLP SOPs, which are critical in terms of that overall system. Okay, next slide. So how do you screen potential CROs? So some of the ways that we found useful for screening out CROs is online research is an easy way. Meet with, and usually it's a combination of things you're going to do, not just one of them. You meet with sales representatives. A good sales representative is actually going to, even if they're not a scientific background or specifically in, say, toxicology background, a lot of them, a good talented one will actually be very, very knowledgeable, especially if they're experienced. They'll just, know what the CRO can and cannot do. And so that can be very beneficial. Scientific conferences and organizations are a great place to do. So if there's an annual meeting, like the Society of Toxicology meeting, all the CROs advertise there, great time to meet with them. Of course, colleague recommendations and just personal experience are awesome too. Okay, next slide. You're going to want to see what types of studies the CRO can do. Can they do pharmacology, PK, toxicology? Do they do stuff that you need to have done? Like I said, are they GLP? Can they do GLP work? Any lab can do non-GLP work, but can they really do GLP work and have all the infrastructure in place to do that? You want to ask, what study functions are conducted in-house versus subcontracted out? It's rare if a CRO at a single site does everything in-house. And I'm talking like, say like histopathology, collecting tissues and, processing the tissues and then sampling them. The bioanalytical work, even if it's the same name CRO, say a Charles River, a lot of times they'll actually outsource or put the bioanalytical or say toxicokinetic analysis at a separate site. So that's actually a separate facility. Oh, sorry about that. And so it's really a different site, even though it's the same CRO. So you just got to figure out, what's going to be done at the place where the animals are versus, stuff that's being, farmed out, and just making sure that you understand the whole setup there. And you want to see, does the CRO have all the capabilities and experience required for the study? They may say they do, but it's good to get the number of studies they've done or the type of work that you're looking to have done. And that's getting into the available historical control data. And then do you want to monitor the study with on-site visits? And then again, you sort of play it by ear. If it's, not a pivotal study, dose range finding study, you probably don't need to go on site, especially if you have experience with the CRO. But if it's a pivotal study that's going to make or break your product, I highly recommend doing at least one on-site visit, if not multiple, depending on the length of the study, just to make sure, one, the CRO knows you're keeping on top of stuff, and then you can actually go see that in person, how things are being done and things are being done well. And then especially even before, you can go like the day before dosing occurs, so you can make sure everyone's on the same page with, what's going to happen on the first day of activities. Okay, you can go to the next slide. Some things you're going to need to think about as you're talking to the CRO is establishing a non-disclosure agreement. The CROs typically want a two-way because they don't want their pricing given out, because that is, sensitive information for them. What's really helpful is to provide a detailed study outline to the CRO so you can get really accurate pricing. So if you just say, I want a 9-month dog study, and you don't give a route administration, the endpoints you want, things like that, you're going to get this generic, quote and endpoints back. And another CRO might give you something different. And so if you give them a really detailed study outline of, what you're looking for in your study and all the endpoints and the dosing, you don't have to have the doses set, but just the dose outline, the number of groups, number of control groups, then they'll give you a lot more accurate pricing that you can compare between CROs. And then, of course, as a company, you're going to need to figure out what is the most important for you, or what the mix of balances is: price, payment schedule, quality, responsiveness, reputation, and figuring out, is the CRO that meets all these, or at least meets them as close as possible? Is that going to drive your selection over other things? Okay, you can go to the next slide. And you can do, once you do your initial screen, you can do a deeper dive, determine if the CRO has specific capabilities or expertise in critical functions to drive the success of your study. Just a few examples here is, do they have a full-time on-site board certified veterinary pathologist to oversee necropsies? Again, is that board certified veterinary pathologist that for GLP studies typically very critical. Not that they're going to be better at their job than a non-board certified. It's just that having that name and stamp of approval is something that FDA is going to really want to see on your study. For this one's interesting is for ocular examinations, especially if it's an ocular drug, you're going to want a board certified veterinary ophthalmologist, and these guys really do exist. And they're very hard to come by, but they're very critical, especially for ocular drugs. Do you need a BSL facility? Do you have, infectious agents? So you need some type of, special handling of the animals and containment. So I'll skip some of those. Do you need complex statistical analysis? Depending on what FDA is asking for, you may need something more complicated in terms of statistical analysis than the lab can provide or is available in their data systems. And so you may need to outsource that part. And the last one is actually pretty interesting. I ran into this personally, that I needed a specific strain of mini pig for a topical product. The original study was run in China, and they have this Bama strain of mini pig, it's called. It's a China specific, and we needed to run the study in the US. And so we literally could not get that strain of mini pig. So that ended up being a real big problem because we needed to run it in a separate lab for various reasons. So that is just things that they can run into that you need to take into consideration. Okay, next slide. And like I mentioned earlier, conducting an on-site audit of the facility. Oh, actually, this is the audit. It's if it's the first time working with the CRO, it is good to conduct an audit of the facility before you actually do any work with them or start designing the study. it could be the same CRO that you worked with before, but it might be a different site. Like I mentioned, like a lot of these CROs now have multiple sites throughout, not just the US, but throughout the world. So if you're doing animal dosing at one site in the past, but now you're going to use another site, it'd probably be good to go see that new site. You want to tour the facility, review their SOPs. And what I think is really critical is establishing relationships with the study director and management, technicians, and scientists. And, just showing, hey, I'm going to be here. I'm going to be involved in my study. And just to have that good communication with everyone. You don't want to ask the lab for FDA warning letters or any kind of inspection reports, because that's really key. You can do a FOIA information request for these things, but it's easier just to ask the lab for them. And then if everything's acceptable, you're going to sign a contract to run the study. However, be aware that legal negotiations can take time. I was working with a small biotech that they claimed they're very nimble, and they had a very nimble legal group that would get things signed immediately. The CRO's legal group was nimble. They had no issues whatsoever signing stuff, but the client's legal group ended up taking, I think it was 8 weeks before they agreed to all the terms. And to me, it's stuff that I considered fairly minor, but it was 8 weeks later that we had to wait before a contract was signed. So just need to be aware of what the legal negotiations can take. Then you want to pay attention, and this is one thing you really do need to pay attention to, especially since, finding a CRO space right now is really difficult, is pay attention to payment schedules, study room reservation cancellation, delay penalties, and other obligations that may incur financial or scheduling penalties. Just as an example is I had one study that I needed to run, but we're waiting on FDA comments on the protocol. FDA kept on delaying, so we couldn't run the study. And luckily it was, I think actually it was a little bit late, that I missed one of these deadlines, and I was going to be incurred like a huge 25% penalty for a study delay. I was able to, since I worked with the CRO a lot, I was able to get that waived as a one-time waiver. But again, it's really important to pay attention to these, especially if things aren't going to go as planned for things that are out of your control. Okay, next slide. Okay, so this next section is just about the importance of active study involvement. So you can go to the next slide. And so you got scientific needs, logistical feasibility, and regulatory compliance. And unfortunately, these three factors do not always align. Just a few examples. The study design may require an excessive number of animals. And so you literally can't put the CRO might not have enough space or technicians to actually run, handle all the animals that you need. Or you may not be able to dose, given the number of animals, dose them or observe them within the restrictions of the protocol that you need to have to meet the regulatory requirements. Like I say, yeah, not enough cages, rooms, techs. Animals, this is another one. The third bullet is animals need to be singly housed for clinical observations. However, animal welfare requirements may require group housing. And there was a study I ran into that the regulatory requirements from FDA required single housing, but the animal welfare requirements it was an outside the US study, were really, really restrictive, and they required group housing almost all times. You had to get the special exception. So that was a really difficult negotiation. Setting the high dose can be problematic sometimes. A lot of people want to match the clinical dosing. However, a lot of tox studies, you need to do a maximum tolerated dose for regulatory acceptance, whereas scientifically, matching the clinical dose may make more sense, but it won't be acceptable regulatory. And then in terms of logistics, the last one, having to dose dogs with 10 large, and I'm talking really large capsules, 5 times per day is just logistically not feasible. So you have to make compromises on that. There you go. Next slide. You could have blood collection volume issues. That's always a problem, especially if the animals, it's not a terminal collection and you're going to do repeated, say pharmacokinetic sampling. You need to figure out blood volume limits and what's acceptable to the IACUC and animal welfare requirements. You may have a required endpoint or analytical instrument that's not validated by the lab, and you might have to do it under, as non-GLP. Ideally, you do it under the GLPs, but you can do it as an exception to GLPs, and hopefully it's acceptable from the regulatory perspective once you submit the study. You may be collecting too many samples during necropsy. In this example is collecting the regular tissues for histopath in this one, but then we need to collect some extra samples for RNA for your gene expression analysis. And trying to collect all those things at one time at a necropsy was, not typical. And we had to really think carefully about the logistics to make sure the RNA didn't degrade during the sample collection time while you're trying to conduct everything under the GLPs and document everything. Okay, you can go to the next slide. And this is where you need really to be involved in the study, and you are going to be involved almost even if you don't want to be involved, because the lab's going to have a lot of questions for you. But even if they don't, like I said, you want to be actively involved and have input on all these things. Where are the animals, bought from, and, the type of, where do you want to get them from? Quarantine and pre-study health assessments you want, animal ID options, animal housing, water feed, environmental controls, a number of types of groups and subgroups. You can do staggered start if there are too many, animals for dosing in a given day. How are you going to randomize to groups? One lab right now is actually, I was actually a little surprised. They used to do a body weight randomization on top of regular randomization. Then they'd make sure body weights were somewhat spread, throughout the different groups evenly. They stopped doing that aspect for some reason. And so I still have them do that part of things. But again, this is things that you're going to want to provide input on. Okay, next slide. You can put on the dosing, what type of in-life evaluations you want. And there's just a laundry list of inputs here, your physical exams. And it's not just the endpoints that you want, but the timing of the endpoints you're going to need to provide input on. Like for ClinPath, for a repeat dose study, a chronic repeat dose study, how often do you want these ClinPath samples collected? And are there dosing, blood volume limitations that are going to limit that too on top of it? All right, you go to the next slide. Again, with terminal procedures, you're going to provide input on all these things: ClinPath, your terminal ClinPath, your gross necropsy, organ weights, tissue preservation, and histopath. And then, like I said, other possible assessments depending on the study requirements. And that one that I mentioned earlier was RNA collection for next-gen sequencing. So that was a really, one-off, unique thing. But again, you might need that for your study, and just figuring out the logistics with that. Okay, you go to the next slide. In terms of the protocol and standard operating procedures, the study protocol must specify all study details unless it's covered by a laboratory SOP. So the SOPs are your first line of how to do stuff. And then your protocol is trumps that. So your protocol really specifies everything. And so if your protocol says to do one thing, SOP says to do something different, you'll follow the study protocol. So it's a combination of these two that you're going to want to look out for, make sure everyone at the lab understands what's driving what there. The study design and protocol, must meet GLP for a GLP study, GLP and animal welfare requirements. The study design should meet applicable regulatory guidance. For example, FDA and ICH both have, ICH is this International Conference for Harmonization. It's a longer name, but they have guidances on how to design different studies for drug approvals. You're going to want to try to have your protocol meet those in your study design. And of course, if you have FDA requesting specific things, so you had to say a pre-investigational new drug application meeting with FDA, and they said, if we wanted this, we say we want 2 control groups in your study, an untreated sham plus the vehicle control, which happens for a lot of topical products. You're going to want to make sure your study design meets that too. So making sure your protocol covers all those things. All right, next slide. Okay, next slide here. So in terms of some of the key determinants of success that I've covered in this presentation are, one, selecting a good lab that meets your fit for purpose requirements, designing a good study in terms of the protocol, active study monitoring, ensuring GLP compliance for a GLP study, and then ensuring animal welfare compliance and trying to balance that with your regulatory GLP and scientific requirements too. Okay, next slide. And just real quick, in terms of the GLPs, I mentioned this before, but they help ensure the study is conducted in compliance with the protocol and SOPs. and that the study is thoroughly documented and it can be reconstructed. So their FDA, basically, if they come and audit the study, they have confidence that the data were collected are accurate. However, the GLP compliance does not ensure regulatory or scientific compliance. So you can have a fully GLP compliant study that does not address a specific request from the FDA. And like that example I mentioned, having 2 control groups for a topical product, you can have a GLP study that has a single control group, fully GLP compliant, perfectly acceptable. But if you don't add that second control group, FDA is not going to accept it. You can have a fully GLP compliant study that does not have the adequate control groups. I used that example, sorry. Okay, let's go to the next slide. One quick update on the GLPs, and I forget when this was. It was 2016 or something. FDA has proposed to revise the GLPs to enhance the existing quality system approach, address multi-site studies. And so multi-site doesn't apply just as a single CRO. It can be or to multiple CROs. It can be to a single CRO, single name, but then they have multiple sites where they're putting samples, as I explained earlier. And so if these things are actually codified in the CFR with the GLP changes, it would address those better than the current GLPs from FDA. And then also better align with other GLPs such as OECD GLPs. So the GLP amendments are actually going to put a little more onerous on the sponsor to keep on track of their studies, which is the stuff I'm talking about. So actually it's going to make the sponsor a little more responsible, which is a good thing. Address multi-site studies, which is a good thing. And then like a better alignment with other GLPs, which I think it's all positive, the GLP amendments that are being proposed. Okay, the next slide. And then the last thing is here is about animal welfare. And so the study protocol will be reviewed by your IACUC committee and approved. They'll ensure that the study complies with, appropriate federal, state, local animal welfare regulations and guidelines. And there's similar requirements for animal welfare protocol review by outside the US CROs. However, animal welfare regulations may be stronger or weaker. And I mentioned that one example is outside the US, they're a lot stronger to the point where to single house the animals for the study, we had to get a one-time exemption from the local animal welfare committee. Okay, next slide. And just be aware that there are a lot of times potential conflicts between animal welfare requirements and scientific or regulatory requirements. And just as an example, under the GLPs, the study director has overall responsibility for the conduct of the study and is the single point of control of the study, which is called. However, under the US animal welfare regulations, the attending veterinarian will make the final determination on animal care and use. So technically, the study director should be doing that under the GLPs, but under the animal welfare requirements of the US, the attending veterinarian makes that decision. So that's technically a conflict of interest there, or a conflict there to be aware of. A lot of times the labs have a SOP that covers this and will address how the study director and animal attending vet will interact. And so it's usually not an issue, but just something to be aware of. Inclusion of humane endpoints, for example, early termination may conflict with scientific requirements. So the animal welfare requirements may require early termination, whereas, to get regulatory acceptance, you may need to continue those animals to the point where it's beyond the animal welfare requirements, and that can be a difficult balance there. Use of sedatives, analgesics, and anesthetics may confound the scientific interpretation of the study, which could, affect regulatory acceptance. And then, like I mentioned before, animal housing, group versus single, environmental enrichment. A lot of the animal welfare requirements are going to require, more group housing and more environmental enrichment, but it may conflict with some of the scientific or regulatory requirements you may have. Okay, next slide. Okay, and then last slide here. So in conclusion, you need to determine how your study and work fits into your overall development plan. Determine which CROs or CDMOs can provide the medical product development services you need that are the best fit for purpose. Once the list is narrowed down, you further screen the CROs or CDMOs, and then you select a CRO or CDMO that best meets the goals of your development plan. You design the study and work so that it meets the relevant scientific and regulatory requirements and try and make the best balance possible there. And then, like I said, I can't emphasize enough, just be actively involved with your study all the way from, selecting the CRO all the way through, final reporting of the study. Okay, so that's it. So thank you for your time.
Okay, great. Thank you, Willie. We did have a handful of questions come in. So the first one, many of these CROs are out of the country. Is the expectation for due diligence to visit those sites, say, that are in India or China, for example?
So Greg, you want to take this one?
Sure. I believe that was actually discussed during your presentation on, I believe, slide 16. So I would say that, since we're still on the COVID pandemic, it would be very difficult to do a due diligence or an on-site audit of those facilities. So what you would need to do is, there's always a representative in the States that would represent the labs in India or in China. And what you need to do is really develop a very strong working relationship with that, either the sales rep or the study director. That would be, your conduit to the labs that are in India or in China.
Okay, next question. Does the FDA have a list of approved CROs, and do these CROs have to reach specific milestones that they must meet to be FDA approved?
So I'll start out. So no, FDA does not have an approved list of CROs because, as I mentioned, the labs are not pre-certified GLP by the FDA. FDA does not assume a lab's doing good GLP work. It's after the study is done, FDA will go and audit them. So FDA does not have a list of GLP approved labs per se. And Greg, you want to add anything to that?
No, nothing to add.
The next question, can you describe issues to watch out for when going from synthesis of small batch to large batch synthesis by CROs?
You want to take that, Greg, or?
Well, I'll just describe my experience about going from a small batch to a large batch. And I always made sure that I had a med chemist and a process chemist involved in the program when we were having things manufactured at a CRO. And they could always answer questions that the CRO may raise in terms of yields or purity of the byproducts that they were generating. So I always made sure that there was a med chemist and a process chemist involved in the program. Anything else, Willie?
No, that's awesome.
Okay, our next question. Typically, do academic institutions want to review the IACUC approval provided by private CROs for NIH grant-supported research?
I can take that, Willie, if you don't mind.
Yeah.
Yeah, I'm not quite sure the question, but it sounds like the lab is running a study that has been approved by the company's IACUC. And if that's the case, even though you have an approved IACUC from the company, since you're running the studies in your facilities, you wouldn't have to get your own institution IACUC approval to run those studies. Now, I will add that if a small company, let me back up, most academic labs or facilities have animal assurance from the Office of Laboratory Animal Welfare, OLAW. There will be a time when a small company has received NIH funding, and usually a small company doesn't have a animal care or animal use set up in their company. So what they would have to do is then identify a lab to run those studies that have a animal assurance, and then they would need to negotiate with that that contract lab and OLAW to set up a interinstitutional assurance agreement. So everything's on the up and up with OLAW.
Okay, thank you for that. We do have one more question, but if there are any other questions that attendees would like to submit, please do so now. And at this time, I'm also going to open the feedback poll in a separate window. So if you wouldn't mind taking a moment to provide your feedback. But our last question I see here, does the FDA do inspections of labs outside of the US?
You want to take that, Greg?
We can take it, Willie.
Yes, yeah, they do. Yes. And I think and I'm actually I forget the exact logistics. I think they have an agreement with the auditing facility, the regulatory body, and the company or country where the CRO is. Well, they'll take that inspection report. I think that's how it works for those types of studies, for a lot of them. Now, if the local country doesn't have an auditing body, then that might be an issue, but I think there's a cross-agency or cross-country agreement there.
And I think I would only add before we end would be that if you are planning or to initiate a GLP study, please have a pre-IND meeting with the FDA. So you're on board and they agree with your program before you initiate very, time-consuming, very expensive studies. So.
Yeah, I will echo that 100%. I've seen companies get away with it, but it's for very rare diseases, very rare unmet needs. So the review division will sort of turn a blind eye, but usually they don't like you submitting stuff without talking to them first. So.
And Carmela Mooney from RTI gave a very nice webinar on that, I believe, last month. So we can link that to this presentation.
Absolutely. All right, everyone. I'd like to just ask one more time if we have any other questions for our wonderful presenters today. If not, I'd just like to remind everybody that the Catalyze application due date is tomorrow at 5 p.m. Your local time. So whatever time zone you're in, 5 p.m. is your deadline. So if you have any remaining questions that we can help answer ahead of that deadline, please reach out to us. You can visit our website, catalyze.org, and send us an email through there. So thank you again for joining our webinar, and a huge thank you to our presenters today.
Yeah, thank you.